Welcome to week 5 of the class Neuronal Dynamics. In the previous 4 weeks, we have looked at neuron models, and all these neuron models were deterministic. There was no notion of noise. Now, in the brain, if experimentalists do recordings, they find a lot of fluctuations. One of the questions we are going to ask, where do these fluctuations come from? Are these fluctuations noise? Now, let's for a moment go back to the full brain, not an isolated neuron, but a full brain with all its different areas, a brain involved in processing of information arising from external stimuli. So here is a recording from a mouse that's awake, sitting in the cage and whisking from time to time. And what we see in this recording is that the membrane potential exhibits strong fluctuations, as we have already discussed in week 1. From time to time, there are action potentials. And one of the questions is the timing of these action potentials also fluctuating? Is this variable? Another question is, where do these fluctuations of the membrane potential come from? Regarding spike timing, let's look at monkey data. Visual cortex is organized in different areas, and one of the areas is called MT or V5, area 5 of visual cortex. Neurons in that area respond strongly to movement stimuli. So if you present a dot pattern on the screen and the pattern of dots is moving, then neurons in MT will respond to this pattern. Now, instead of presenting just a coherently moving object, one can think of presenting random dot motion. So there's a random configuration of dots, they move in some directions, and if the very same random dot motion pattern is repeated several times, then you see in the first trial spikes arrive here and there, and there, and there, and there, and there, and there, and there. Now the same pattern is repeated a second time. Spike timing is different. Spike timing is different at every different trial. If you average over several trials, then you find that a lot of activity occurs here and again here, but then there's a pause and there's more activity coming here and there. So on a time scale of 1 second, timing looks fairly reliable. This neuron emits spikes reliably in response to such a random dot motion. However, if we zoom in on the timescale of milliseconds, then we see that there are a lot of fluctuations of spike timing between one trial and the next. Now, this is monkey data. There's also data from humans. Human patients who suffer from a severe form of epilepsy, a form of epilepsy that cannot be treated by pharmacological means, then these human patients often have to undergo a surgical treatment. Now, before the operation, an electrode is implanted in the brain, and the electrode stays there for several days while humans are awake. You can talk with the human patients, you can present pictures, and so this is recordings from an electrode, and we find that neurons close to this electrode respond strongly to images of, say, the Sydney Opera House. And the same neuron respond in trial number 1, in trial number 2, in trial number 3, in trial number 4, and so forth. And if you average across different trials, you see a strong peak in the number of spikes during the presentation of the image. The same neurons also respond if you just present the name Sydney Opera House. And in fact, they continue to respond, indicating memory. Opera House evokes some memory. Neurons, or this one neuron, sitting close to the electrode, responds for a prolonged time. However, between individual trials, timing does not timing of spikes does not look reliable. Now, while this neuron responds to Opera House, it does not respond to the Pisa Tower, but there is always some spontaneous activity going on which is not repeatable between one trial and the next. So this is data in vivo. In brain slices of mouse cortex or rat cortex, you can isolate a neuron and inject a specific time-dependent current into the neuron while recording, say with a second electrode, the membrane potential. Now, if the very same fluctuating input current is injected several times, then you see that the neuron responds, but it responds each time at a slightly different moment. In other words, spike timing is not reliable. So, spike timing is not reliable. The neuron models we have discussed in the first 2 weeks were deterministic models. If you repeat the same stimulus, they respond the same. In the brain, we find fluctuations, fluctuating membrane potentials, fluctuating spike times. So the question arises, well, what's the meaning of these spikes? What does that mean for information transmission? How can neuron encode information in the presence of these fluctuations? Are these fluctuations noise? Can we model these fluctuations? Now, these are the questions that we will address in the next few lectures. Please stay with us.